To determine the geographic origin of the virus associated with an outbreak of the Drafa virus, we conducted a phylogenetic analysis of all native sequences together with those of the current disease outbreak. We computed a neighbor joining tree from the nucleotide sequences, which we then visualized with our tree visualizer. The tree visualizer is linked to a matrix viewer containing the alignment. The tree visualizer allows interactive navigation in the tree, selection of nodes or edges, which are then highlighted. The tree shows that all virus sequences from the disease outbreak are very closely related and cluster monophyletically. This proves that all strains from the current outbreak have one common ancestor. The native strain that shares the lowest common ancestor with all strains of the current outbreak is the strain from Nigeria B, so the answer is Nigeria B. After performing a phylogenetic analysis of the nucleotide sequences restricted to the sequences of the current disease outbreak, we imported the resulting tree in the tree visualizer. In the matrix viewer, we can sort the IDs and select them individually. The corresponding nodes are simultaneously selected and highlighted in the tree. We selected the three IDs with label 583, 123 and 51. 583 is the label of Nikolai. It can immediately be seen that 123 is much closer in the tree to 583 than the sequence with ID 51. Therefore, the answer is that patient 123 most likely contracted the virus from Nikolai. To answer this question, we worked in the matrix viewer. By showing the alignment and the attribute simultaneously, a compound view of the genotype and the phenotype is provided. In the matrix viewer, the cells of the matrix are colored by nucleotide or attribute value. For this, disease characteristics were linearly mapped to integers 0, 1 and 2. Columns in the matrix viewer can be moved, sorted and hidden. We first noted that many positions in the alignment are perfectly conserved, that is, all strains have an identical nucleotide at that position. Since conserved positions cannot contribute to virulence of a mutant, the tool can automatically hide these columns. The same can be conducted for columns with only one mutation. Next, we sorted the rows by severity. There are three groups representing low, medium and high severity. We select the rows of each group. Rows can be used to compute the consensus sequence of all selected sequences. The nucleotide of the consensus sequence at a given position is chosen as the one with largest frequency. Opacity is mapped to the relative frequency of the nucleotide in the consensus. The most prominent correlation of columns with the gradient of the severity attribute column are those with decreasing opacity, as these indicate increasing mutation rate in the respective group. We note that only two columns, 223 and 269, show this pattern. There are several other columns that either show a reverse opacity pattern or even different letters. Here we suspected that some of these columns are related to overall virulence. Therefore, we now first answer question number 4. We computed an additional attribute by summing all symptoms for each patient. The entries of the sum statistic range from 1 to 8. We sorted all rows according to the sum statistic and consecutively computed consensus sequences until only 4 phenotype levels of overall virulence remained. We see 6 prominent columns. To identify which of these are correlated mutations, we computed the pairwise mutual information. Altogether, our 3 top mutations for overall virulence are columns 842-946 and columns 2279 and columns 790-161. The third mutation for severity is position number 109.